Hi, I'm Professor James Cairns, or as everyone calls me, Ajahn Jim. And I want to help introduce the work in our lab, research that we're doing in our lab. Our main interest is how molecules function, and in particular, our molecule of choice are proteins, and particularly carbohydrate active enzymes that can catalyze a reaction related to carbohydrate, because this is very important for both how living organisms survive and also for helping us to manipulate them and do things like biomass transformation. Okay, so we're interested in how enzymes work. So you can see this is the structure of one enzyme that we were the first in the world to solve the structure for this, a member of the family of enzymes. This is just a cartoon picture, but you can see the ball and stick pictures of some glycerol from the cryoprotectant for our X-ray crystallography and an inhibitor in the active site. And when we publish this, other companies are interested in inhibitors because this enzyme is related to human enzyme that is defective in some neurological disease and might be a target for drugs for other diseases. So as soon as we published, a company in the US out of Harvard University was interested to contact us and work with us on this enzyme. So even though we're interested in very basic ideas, we have applications to high-level industry that will be important. So again, basically, we're interested in knowing in the bacteria like this one, or we can make a model of the human enzyme based on our structure. How does that enzyme work in the human? So sometime we work on enzymes from plants, and then we want to go in and see what metabolites are affected by that enzyme by overproducing or underproducing it, and what metabolites does it work on. So we're involved in natural products extraction, like some organic chemists. We also sometimes want to synthesize the substrate or synthesize inhibitors for our enzymes, and some of those can be applied to things like possible treatment for COVID-19 or other diseases. So we are working on that, and we have chemical synthesis in our lab as well. But most of our work is in the biological, producing these enzymes like this one in a bacteria usually, or a yeast maybe, and then trying to manipulate the active site, either to engineer it to do a new kind of reaction for us or improve something like biomass degradation for us. Sometimes we had to go into the plant or human cells, and then we extract, say, lipids and use mass spec to tell us which lipids are there in metabolomic to see if we have too much of an enzyme or none of the enzyme, what happened to those metabolite levels. So we're working at many, many different levels in our lab, but it's all about protein function. What do the proteins do, and how do they do their function?
Hello, my name is Wang Meng. I come from China. I'm the fourth year PhD student in Ajahn Jim lab. Welcome to the JKC Biochemistry Lab tour. สวัสดีครับ ผมเป็นชื่อของหวังมาจากประเทศจีนครับแล้วก็ไปเที่ยว JKC Lab ครับ This is our working bench, and we have the bottles, tubes, or some pipette. Here's Miss Chamebon. It's our research assistant. She's doing the enzyme kinetics. Hello.
Hello.
Dr. Manur is our senior PhD student and going to finish her surfing PhD life. No, I shouldn't say like this. I just say hi to.
Hi everyone, my name is Luna. I work on pan beta glucosidase.
My name is Mariganda Shanti. I am coming from India and I am working under the James Achan team, and my work is organic synthesis and total synthesis of sphingosine.
Beside Dr. Maniganda is our new British guy from Myanmar. It's lab2n. Yeah.
Okay, let me introduce myself. My name is Tom Win and I come from Myanmar. So today my lab is especially for the extraction of phenolic compound for rice. In here I would use a lot of rice sample.
Hello, my name is Karuna. Okay, I am from India. My PhD work is functional and then characterization of enzymes from plant. For that, I am using the wild type, and then mutant plants. I am using Arabidopsis as a model organism. For growing the plants, we are having the plant tissue culture lab. After this, we can explore the facility of our lab. So, thank you.